Hi, everyone. My name is Peter Fung, and I'm the Senior Manager of Customer Marketing here at Nanotemper Technologies, and I'll be your moderator for today's presentation. The topic we're going to touch on today is therapeutic antibodies. So as many of you know, therapeutic antibodies have become one of the most predominant classes of new drugs being developed today. They've been used to treat a wide variety of different diseases, including cancer, autoimmune disorders, as well as many metabolic and infectious diseases. So clearly, there's a continuing need for scientists to more efficiently identify, characterize, and develop new and novel therapeutic antibodies. So with that, I'd like to introduce today's speaker, Dr. Lindsay Dawson. She got her PhD in chemistry and biochemistry at UC San Diego, and she's currently an application specialist here at Nanotemper Technologies. Just to share a couple of fun facts about Lindsay before we get started, besides being an amazing scientist, she's an avid runner. She even qualified for the New York City Marathon. And Lindsay is a total foodie and wine enthusiast, so I think you'll really enjoy today's presentation. The title of her talk is Measuring Stability to More Effectively Select Therapeutic Antibody Candidates. Take it away, Lindsay.
Thanks so much for the introduction, Peter. And just to kind of reiterate what Peter said about the importance of these therapeutic antibodies, I wanted to share with you some of the top pharmaceutical sales from 2018, where we can really highlight how impactful these therapeutic antibodies have become. So we can see that not only our top seller, Humira, is a monoclonal antibody, but we also have 7 other monoclonal antibodies in this list. Additionally, we have over 570 monoclonal antibodies at various clinical phases, 62 of which are now in late-stage clinical studies. So clearly, we can see how important these therapeutics are, and really that we need effective tools to make them develop faster and be more efficient and deliver them to the market. We also know that the discovery of these therapeutic antibodies is extremely costly. It can be very laborious, and oftentimes the process is fraught with failure. And we know that by characterizing specific attributes of these antibodies up front can really help you address these issues of high cost and high labor. So we know that thoroughly characterizing the stability of your antibody candidates all the way from early development out until clinical release is one really impactful way that you can increase that efficiency of getting that drug to market. So let's just highlight quickly where stability can be monitored throughout this drug development pipeline for these therapeutic antibodies. In target identification and validation, we know that QC of the antigen is of the utmost importance in order for you to develop a valid target and validate that therapeutic antibody. Moving over to lead discovery, we know that you then need to purify this target antigen-specific antibody. And in order to do that in a reproducible way, you need to have an effective tool that can help you measure stability. Additionally, when we move over to modification of these antibodies, we know the direct impacts these modifications, such as antibody drug conjugate development, affinity maturation, or even just looking at antibody engineering when you're working with Fab or scFv fragments. All of those things have a substantial impact on the overall stability of the antibody, and you need a tool that will help you precisely characterize those changes and allow you to make decisions based upon the changes of the stability. Lastly, we have bioanalytical characterization, where we know thermal stability, aggregation propensity, and glycosylation patterns are all very important parameters to characterize thoroughly as you're developing these drugs. Sort of towards the end of the development process, we have antibody manufacturing and formulation to fill and finish. And we know with manufacturing, a lot of times the manufacturing can happen across sites. And you need, again, these special tools that will enable you to make these effective decisions in order for you to continuously generate reproducible, quality-controlled antibodies throughout the manufacturing process from site to site. And then lastly, formulation, looking at a high throughput method to help you screen quickly through those formulations, as well as being able to subject the formula to then stress tests such as freeze-thaw cycles, or looking at long-term stability. Again, monitoring stability throughout those processes is of the utmost importance. So this is really where we come to how you can measure those stability parameters throughout the development process, and how the Prometheus with nanoDSF technology can help you improve efficiency throughout the antibody development process. So just to quickly highlight the benefits of this instrument, we have measure under native conditions. A broad detection range really means that you can use this instrument throughout the development process, from the target identification selection all the way out to the formulation, as we mentioned. And then ultra high resolution means that you're going to be able to capture those individual domain unfolding events. Also, we'll talk about this in a few slides, but simultaneous detection of protein aggregation is really something that can be impactful in making effective decisions. And then using low amount of sample is really beneficial throughout the entire process, so you're not losing precious sample. And then lastly, but certainly not least, is offering a flexible throughput with automation options available. So this is why we'd really encourage you to join our growing community of Prometheus users. And we're just highlighting a few of our academic and industry accounts that we work with on a daily basis, and who are already using this technology and say that this technology is the new gold standard for characterizing protein stability. So let's see why that's the case. I want to start out by introducing a customer that we have who really speaks to an all too familiar case of an issue that comes up a lot, that really needs to be caught early on to avoid costly down the road poor decision making in this development process. So Dr. Alan Kutach from NGM Bio says that they use the Prometheus to evaluate molecules at early stages, taking advantage of its sensitivity to avoid costly scale up of candidates that will not move forward. Additionally, because they could compare all aspects using the Prometheus, it revealed an unanticipated interaction between domains of a non-antibody fusion protein that rendered the molecule unmanufactured despite its robust activity in functional assays. So they really could have gone down this rabbit hole in pursuing this candidate until they were able to get this holistic overview of these unanticipated interactions using the Prometheus to make a decision to not go forward with this candidate. All right, so let's quickly talk about what nanoDSF is and how the Prometheus uses nanoDSF to measure simultaneously conformation and colloidal stability. So nanoDSF stands for nanodifferential scanning fluorimetry, and basically what it is using intrinsic fluorescence of a protein to monitor the unfolding of that protein to characterize, using a TM value, the stability of that protein. So unlike DSF, which uses dye, again, we're using this intrinsic fluorescence, and that fluorescence is measured from our tryptophan and tyrosine amino acids from our protein. So essentially what we do is we start out with a folded protein we can see over here on the left. And basically we expose that protein to increasing temperature or chemical denaturant. And as that protein is exposed to that increase, it starts to lose its overall three-dimensional structure. As such, the tryptophan and tyrosine residues go from a hydrophobic core of the protein to an exposed hydrophilic environment. And that change in environment also changes the emission properties of these amino acids. And so what we're monitoring is that change in emission properties, which then gives us this unfolding profile where we're monitoring the change from folded to unfolded. And then about the midpoint of this folded to unfolded profile, we're able to characterize the TM or melting point of your protein, which can then be used to quickly screen through different buffer conditions or different variants of your protein to evaluate the stability. I mentioned that we're simultaneously detecting colloidal stability, and the way that we do that is we look at aggregation using our back reflection technology. And back reflection technology is a technique that's based on scattering, and essentially what we're doing is we're collecting a loss of light that's been shined through our sample. So this principle is demonstrated over here on our left, where we're simultaneously collecting this incident light that has been shined down on our sample. And we can see that as the sample starts to aggregate, more light begins to scatter, and so we lose the light that's being measured back into the detector. And this is, again, happening at the same time as the detection of the fluorescence on the same sample. And so what the data looks like is what we see on the right-hand side, where it looks similar to an unfolding profile, but really what we're trying to extract from this information is what we call the onset of aggregation. And this information is useful in characterizing your proteins going forward as a parameter that you want to keep your samples under to keep them from aggregating, as aggregation is often irreversible. All right, so let's talk about some examples where you can more effectively monitor therapeutic antibody stability using our nanoDSF technology. So one example is enzymatic fragmentation of antibodies. We know that this process traditionally involves optimizing the ratio of enzyme to antibody, and then also optimizing the amount of time that you're incubating the digestion. So traditionally what you would do is run the samples on an SDS page gel. There's absolutely nothing wrong with that, but we know that that can be time consuming. So what we did was we chose one parameter to quickly monitor and characterize, which was our second TM of this trastuzumab antibody. And what we did was we took 4 different enzyme concentrations at 3 different incubation times. And the data that we want to focus on is for the 1 unit of enzyme concentration. And we can see from the 3-hour to the 6-hour to the overnight, we get a change in that TM value. The other thing to notice here is basically the reproducibility of the replicates that were run. So we can see that the overnight sample really has the lowest amount of standard deviation from the mean. And also it has basically the highest TM value. So from being able to stick this sample into the Prometheus, do a quick 10-minute run, look at the data, we can select the overnight incubation at 1 unit as being our optimal conditions. Another really common study for these therapeutic antibodies involves deglycosylation. So we know that this is traditionally used to simplify protein analysis, remove sample heterogeneity, and also analyze the direct effects of deglycosylation or glycosylation on protein activity and stability. So what we did in this study was we took etanercept or Enbrel, and we essentially removed the O-linked glycans and the N-linked glycans. And then we did another sample where we removed both glycans just to see what impact on stability this had. And I just want to quickly go through this data here to give you an idea of how fast you're able to then analyze the impact on stability by carrying out these various deglycosylation events. So we'll start by looking at the purple trace, which is just the etanercept alone. And we can see 2 distinct unfolding transitions. And then on the bottom is the scattering data or aggregation data. So we see that it's a flat line and there's absolutely no aggregation present in this sample. Upon removal of those N-linked glycans, we can see actually a significant destabilization of the melting temperature of that primary peak in the antibody. And then also we can notice that removing those N-linked glycans still doesn't have any impact on the aggregation, and the antibody is still stable throughout the experiment. When we go ahead and remove those O-linked glycans, it's interesting here because we actually see a slight stabilization of the melting temperature for this antibody. But then when we look at the scattering data below, we can actually see that the antibody has less colloidal stability above, let's say, 82 degrees as it begins to aggregate. And then lastly, when we remove both glycans, we can see a substantial decrease in TM, really destabilizing that protein, and then also onset of aggregation, which is happening roughly around 50 to 55 degrees and continuing on throughout the experiment. So really removing both of those glycans is not beneficial for your studies, and you can make a call on that and not have to pursue any work with those samples, as you know they're probably going to be pretty tricky to work with. And then lastly, I just want to highlight a case study from a paper that was recently published in Analytical Biochemistry out of Amgen. And what they did was they wanted to validate the Prometheus for characterization of therapeutic proteins. So their overall goal of this study was to compare specificity, sensitivity, accuracy, precision, limit of detection, and resolution of protein samples using either differential scanning calorimetry or the Prometheus using nanoDSF technology. In addition, their overall aim is to identify a method that will generate reliable results within the precision and reproducibility that's often required to select these lead candidates. And they also wanted something that was amenable to existing workflows and that could be used on a wide array of target candidates. Lastly, they also wanted to provide both sample and time saving going forward throughout this selection process. So let's just look at a few of the data points that they collected. And we'll start out by looking at specificity and accuracy. So specificity for them meant that they wanted to understand if the nanoDSF technology is giving them information specific to their target molecule, and anything additive to the buffer wasn't having a background signal or impacting the data that they were getting out. So what they did here was they just ran the formulation buffer alone, and then they ran the bispecific T-cell engager or BiTE molecule in comparison. And we can see a nice flat line for the formulation buffer, indicating that there's no background signal from that, and then a really nice sharp peak for our BiTE molecule, indicating we're getting a specific signal just for that BiTE molecule. Accuracy for them meant comparing this nanoDSF data to their existing DSC data, as it's the industry standard to use DSC. So what they wanted to do was understand that for all of their samples, are the TM1s and TM2s of their antibodies the same as the data they're getting out from DSC? And for all of the protein samples that they ran in this study, I would highly encourage you to check it out, because a lot of them were within a very narrow window of similarity to each other, indicating that the data they were getting out from nanoDSF was just as accurate as the DSC data. They also looked at precision, resolution, and limit of detection all in one study. So let's break that down. What they did was they developed this parameter called repeatability, where they examined 7 replicates of the same sample at various concentrations to look at precision, resolution, and limit of detection all at the same time. So let's just highlight this monoclonal antibody 6 that they looked at 4 different concentrations, and we can see a really nice example here where the repeatability is less than 0.1 degrees, meaning that essentially the error within those different samples was less than 0.1 degrees between replicates. And then also highlighting limit of detection here, looking at the same antibody at various concentrations to make sure they're getting the same information at low concentrations all the way up to high concentrations. And then in terms of resolution, they wanted to understand if they were able to get those individual unfolding events for each domain of the antibody. So looking at the Fab, CH2, and CH3 domains individually, and then again looking at the comparability and repeatability of those experiments for those domains. So overall, they were able to find that compared to the current industry standard of DSC, the Prometheus enabled the thermal stability analysis for a much wider concentration range, consumed considerably less material, and provided significantly higher throughput. And that was their overall aim in the original introduction of the paper, and that was something that they were able to establish by the end of their experiments. And just to summarize their conclusions, they said, based on our qualification results, we conclude that the Prometheus is suitable for thermal stability analysis for all studied protein modalities and its applications in comparable studies. So a really impactful statement. And again, I would definitely encourage you to go check out the paper. There's a lot of useful information in there. All right, just to wrap up, I hope that I've made the case to you that Prometheus really is the new gold standard that precisely characterizes protein stability. And the reason for that being is that we're able to simultaneously measure this conformation and colloidal stability of your proteins. You're also able to perform label-free detection in a very small amount of sample, measure over a wide concentration range, and also look at buffer and detergent independent samples. And lastly, it also offers that flexible throughput and it's automation friendly. And I also want to point out that NanoTemper doesn't just offer solutions to characterize protein stability. They also offer everyday solutions for protein quality checks using our Tycho. We have an entire line called our Monolith that's dedicated to measuring molecular interactions. And then lastly, we also offer high throughput solutions for screening and measuring binding affinities. And if you have any further questions, my contact information is listed below, and you can also visit us on our website at nanotempertech.com.
Thanks, Lindsay, for a great presentation. We had a few questions that came up during your presentation, if you don't mind addressing.
Absolutely.
Great. So during the development or even the formulation phases, once you have this collective information that you've gathered, how do you go about selecting the best candidates? Do you have any tips or suggestions?
Yeah, it's a really great question. And I would say it's definitely going to depend on your overall goals and what you've ranked in prioritization. But really, the best solution is going to be looking at all the data that's provided to you. So, collectively looking at TM, onset of aggregation, as well as onset of melting together. And our software for the Prometheus actually offers a really nice way for you to quickly look at those parameters and be able to select candidates based on the analysis that's carried out by the software.
Okay, great. And also, can you automate the screening for thermal stability on Prometheus? If you're trying to test maybe a lot of different candidates or different conditions, is that possible?
Yeah, absolutely. So as I mentioned, we have flexible formats available, and one option is to couple the instrument with a robotic auto sampler, which allows you to screen through 1536 roughly sample conditions in one single run. So really being able to ramp up those selection criteria and run through those formulation or variants in a quick manner using the robotic auto sampler.
Great. And one last question is, when you're looking at both this TM and the onset of aggregation, is one parameter more important to prioritize? What's your feelings on that?
Yeah, I would say no, definitely not. But again, it just depends on how you're analyzing your data and what's important to you to prioritize. But really, as I mentioned, the best thing is to have this holistic overview in looking at all of the parameters combined and making a decision based on the collection comparison versus individual data points.
Great. Thanks again, Lindsay, for a wonderful presentation. And thanks to our audience for taking the time to join us. Again, if you have any questions, please send them out to Lindsay at the email address that's shown on your screen, or feel free to visit our website at nanotempertech.com to learn more about the different products that we have to characterize proteins. So thanks again, everyone, for taking the time to join us, and we hope to hear from you soon.
Thanks.
Bye-bye.